Hello, and welcome to our Neuro project. Today we're going to go over Alzheimer's. The students who worked hard on this project are Allison Stevens, Charles Penning, Michelle Lim, and Bita Beheshti. First, we're going to go over the gross anatomy. In the brain MRI of patients with Alzheimer's disease, there are differences in the gross shape of the corpus callosum and its subregional areas when compared to the brain of an individual with an age and gender matched healthy brain. To the right, you can see in the figure the corpus callosum. The corpus callosum is a broad band of commissured nerve fibers joining the two hemispheres of the brain. The brains of patients with Alzheimer's differ from normal patients in that they have a more convex shape and a reduced area of the corpus callosum. In this figure, you can see what we mean by the smaller corpus callosum. In figure A, the corpus callosum is bent in a downward angle and shrunken. This is a scan of a brain with Alzheimer's disorder. In figure B, the corpus callosum is smoothly rounded and is equally distributed. This is a scan of a healthy brain. The hippocampus. One of the first areas damaged in the brain in Alzheimer's disorder is the hippocampus and its connected structures. Damage to these areas would make it harder for someone to form new memories or learn new information. The hippocampus is needed for the retrieval of newer memories. This is why someone in the early stages of Alzheimer's may remember a childhood memory but struggle to remember what they did earlier that same day. As Alzheimer's spreads throughout the brain, lobes begin to shrink and atrophy begins. As the cortex continues to thin, memories from longer ago are lost. Damages to the left hemisphere of the brain is linked with problems with somatic memory. Somatic memory is general world knowledge that we have accumulated throughout our lives, and language. This causes someone with Alzheimer's disorder to struggle to find the right word for someone or something, such as a color, letter, or name. Damage to the brain. Damage to the temporal lobes make it harder for someone with Alzheimer's to recognize familiar faces and objects. This is why someone with Alzheimer's often forgets familiar faces of close loved ones. However, since the pathways for vision and hearing are separate, they may still recognize the person once they have heard them speak. Damage to the right parietal lobe leads to a person having problems with depth perception in 3 dimensions, such as stairs. Those are a very common difficulty with people with Alzheimer's. As the damage spreads to the frontal lobe of the brain, the person may begin to struggle with decision-making, organizing, and planning. Despite all these losses, though, many abilities are retained, especially those acquired long ago. Patients often remember skills like playing piano because those memories are retained the longest. The limbic lobe is a complex set of 3 C-shaped structures containing both gray and white matter. It lies deep within the brain and includes portions of all the lobes of the cerebral hemisphere. The limbic system consists of a number of structures, including the fornix, hippocampus, cingulate gyrus, amygdala, the parahippocampal gyrus, and parts of the thalamus. You can see all of them in the figure to your right. Alzheimer's disease is a chronic progressive dementia of unknown origin. Early on, patients develop memory impairment which eventually progresses to global intellectual dysfunction. Degenerative changes in the limbic system likely have a role in the genesis of neurodegenerative diseases like Alzheimer's disease. The hippocampus, which plays a big role in long-term memory, is one of the first areas that's affected by Alzheimer's. As the disease progresses, damage extends throughout the lobes. The nucleus basilis of Maynard is a group of neurons in the substantia innomitata of the basal forebrain, which has wide projections to the neocortex and is rich in acetylcholine and choline acetyltransferase. In Parkinson's and Alzheimer's disease, the nucleus basilis undergoes degeneration. It has been well established that neuronal loss within the cholinergic nucleus basilis of Maynard correlates with cognitive decline in dementing disorders like Alzheimer's disease. I guess it is demanding too. Motor system tracks. Although Alzheimer's begins in the limbic system, its effects are not limited to emotion and memory changes. As the disease progresses, widespread neuronal damage significantly affects motor system tracks. Alzheimer's causes motor apraxia. First, sophisticated motor skills are impaired. These include complex skills such as relating to a job. Later, instinctive functions may begin to become impaired, such as chewing and walking, and in general, motor impairment is considered a late-stage symptom of the disease. Primary motor cortex is responsible for integrating muscles in meaningful ways. During the course of Alzheimer's, though, it is affected by neuronal degeneration and formation of plaques and tangles. Why doesn't degeneration in the primary cortex show until late stages of the disease? Research suggests that the motor cortex is able to reorganize itself in order to compensate for neuronal damage. The corona radiata and the internal capsule are both made up of white matter. Alzheimer's is considered a gray matter disease, with the majority of the damage involving nerve cell bodies and dendrites. However, damage to the brain's white matter may be an early sign of Alzheimer's. White matter damage plays a role in how the disease strikes and progresses. As the white matter relays motor information, damage would impair efferent signal transmission throughout both cortical and subcortical structures. As Alzheimer's also causes a degeneration of subcortical gray matter, the basal ganglia and thalamus are a part of the impaired motor pathway of the disease. Shrinkage of the basal ganglia would interrupt the spread and efficiency of efferent signal transmission due to the interruption of normal production and regulation of neurotransmitters. Shrinkage of the thalamus would impair coordination of efferent motor signals before they are relayed down to the brainstem. Generally, the motor problems related to brainstem damage are related to late-stage Alzheimer's. As the brainstem regulates gross motor skills and autonomic functions, these problems can include difficulty swallowing, erratic blood pressure, or difficulty breathing. The ventricles of the brain contain a cerebrospinal fluid. Cerebrospinal fluid cushions the brain, circulates nutrients, and removes waste. Overall, brain tissue shrinkage in Alzheimer's disease causes the ventricles of the brain to appear larger than normal. Ventricles enlargement occurs also in cases of mild cognitive impairment, but with Alzheimer's, it occurs 60% more rapidly. Ventricle enlargement is such a core characteristic of Alzheimer's disease that using ventricular volume as a measure of disease progression is supported by multiple studies. Evidence supports that ventricular enlargement differences may actually be characterized by genotype. The images to the right is a comparison of hippocampal and ventricle volumes between Alzheimer's disease patients. Normally elderly individuals and individuals with mild cognitive impairment. The line in the blue box in the median volume, the cross in the mean volume, and the whiskers display the ranges of the volume. Neurons and physiology. Changes in the brain Although early stages of Alzheimer's disease are not easily identified, some early signs can include the following: abnormal amyloid protein deposits, plaque substances on dendrites and axons, tau protein tangles on the microtubules of the neuron. Abnormal amyloid protein deposits and tau protein tangles contribute to damage within healthy neurons and impair proper neuronal functioning. There's 3 abnormalities found within the brain. Amyloid plaques, typically found within spaces between neurons. They contain beta amyloid protein fragments that originate from a larger protein called amyloid precursor protein, APP. When the body produces beta amyloid protein in excessive quantities, clumping occurs. This is known as the formation of amyloid plaques. Amyloid plaques impair neuronal connections and communication between neurons. Neurofibrillary tangles. That's the second abnormality found within the brain. They're found within neurons. They contain proteins called tau, which clump together to create tangles. They block nutrients and molecules from entering the neuron cell body, axon, and dendrite. Tangles disrupt microtubule networks and create blockages in synaptic signaling within neurons. The last abnormality found within the brain is a loss of cells and neuronal connections. As abnormal deposits of both amyloid plaques and neurofibrillary tangles increase, synaptic connections within neurons decrease. Axons cannot function properly and begin to atrophy, and tissue shrinkage occurs within the brain. The role of neurotransmitters with Alzheimer's disorder. Signals that form memories and thoughts move through an individual nerve cell as a tiny electrical charge. Nerve cells connect to one another at synapses. When a charge reaches a synapses, it may trigger release of tiny bursts of chemicals called neurotransmitters. The neurotransmitters travel across the synapses, carrying signals to other cells. Alzheimer's disease disrupts both the way electrical charges travel within cells and the activity of the neurotransmitters. Acetylcholine, a neurotransmitter essential for processing memory and learning, is decreased in both concentration and function in patients with Alzheimer's disease. Sensory system and tracts. As neurofibrillary tangles spread from the hippocampus throughout the whole brain following a centrifugal movement, the process causes atrophy in cortical structures, causing sensory dysfunction. Olfactory impairment. Neuronal loss in the form of amyloid plaques and neurofibrillary tangles in the olfactory region next to the hippocampus can cause impaired odor identification, odor detection threshold, and odor recognition memory. The olfactory pathway is the most direct path between the hippocampal region and the external environment, which is the outside of the body and through the nose. Olfaction, the sense of smell, begins with sensory neuron input to the olfactory bulb. Then, scent stimuli is decoded in the piriform cortex, and ultimately, information is sent to neurons in the hippocampus. The entorhinal cortex and the hippocampus are altered in response to odor stimuli or odor-dependent tasks. Olfactory impairment is prevalent in Alzheimer's disease pathway and is present in the peripheral and central olfactory neural network in most Alzheimer's disorders patients. Next, you have visual impairment. Amyloid protein pathology has been shown to be present in most, if not all, segments of the visual pathway. The peripheral nerve components of the visual pathway, in particular the retinal ganglion cell axons and the optic nerve fiber layer, develop cell death and axon degeneration in patients with Alzheimer's disorder. Posterior cortical visual areas in the occipital lobe can be substantially affected with Alzheimer's disorders pathologies in the early stages or prior to the onset of dementia. Both the sensory perceptual components and the cognitive components involved in motion perception under optic flow are affected. Central visual dysfunction or cortical blindness is an important part of functional impairment in Alzheimer's disorder. Hypoactivation due to atrophy in the posterior visual association areas is strongly associated with the navigational impairment or visual spatial disorientation in Alzheimer's disorder. Hearing impairment. Alzheimer's disorder pathology is found in the central areas of the auditory neural pathway in most AD patients, but not in the peripheral ear and cochlear structures. Both plaques and tangles have been found in many major components of the central auditory pathways, including the superior temporal gyrus in the cortex, central nucleus, and the dorsomedial nucleus of the inferior colliculus. Brain structures responsible for processing sound and speech atrophy and shrink, including the superior, middle, and inferior temporal gyri. AD patients can acquire sensory neural hearing loss and central hearing deficits. Recruitment of regions in the frontal, temporal, and parietal lobes to maintain auditory speech processing may explain the general preservation of language comprehension that is seen even in individuals with severe AD. Taste impairment. Taste identification is also impaired in the early stages of late-onset AD. This may be due to changes in the innervation of the taste buds. After ingestion of a tastant, which is a substance that stimulates the sense of taste, taste buds are activated and gustatory information is carried to the facial nerve, which is 7. Glossopharyngeal nerve, 9, and vagus 10 nerves to nuclear regions of the brain for processing. AD patients show significant declines in the innervation of taste buds located in the foliate and circumvallent papillae of the tongue at the level of the taste bud. Brain-derived neurotropic factors, BDNFs, if you will, are essential in the development of papillae taste buds. Amyloid plaques reduce the transcription of BDNF in the hippocampus, thereby altering taste sensations. AD patients show losses in the ability to identify the 4 basic tastes: sweet, sour, salty, and bitter. And that, my classmates, is our presentation. Thank you.